Servus aus Berlin. Hi, my name is Matthias Rillig, and today I'll talk to you about how to critically evaluate a research paper. Now, I have to preface this with the remark that this is very hard, and it is a skill that needs to be honed over time. We critically evaluate research papers every Monday in our lab meetings, and every time the group as a whole comes up with things, points to criticize or think about in a paper that I hadn't thought of on my own. And so this illustrates that there's so many things to think about when reading a paper, and therefore this is far from trivial. But it's also a very important skill, because this is how you learn to critically evaluate information. And later on, if you're already a reviewer for journal articles, you will be asked to do this in a more formal sense, of course. But even if you're not a reviewer, you will still need to critically evaluate information that's out there before you use it. And so here are several points. The first point is, does the study design match the question that is being asked? That's a very important point, and it has several categories that are important to consider. First of all, is it the right scope in terms of study object? To give you an example, if the question that is being asked is a general question about, let's say, invasive plant species do this and that. And then in the paper, what is being actually presented are data from one particular invasive plant species, then clearly the actual design of the experiment and the question, the level of generality of the question, do not match. And therefore this is already not basically adequate, because in order to make a statement about, let's say, invasive plant species, you need to replicate instances of a plant being invasive. And this goes, of course, for other examples as well. So basically the first point is the study object matching really the question that you asked? Is it the right scope in terms of object? The next question on the list is it the right scope in terms of space and time? So is this a global study? Then you should look, is the coverage of the sample sites really truly global, or are we talking about 12, 15 sites that happen to be distributed globally? Then maybe you can say something about various biomes to the extent that they have been replicated, but you cannot say anything really about a global pattern per se, because that requires a lot more sample size. And if the question is about seasonality, maybe you should then look for more than one season having been replicated, or is this just one time course through one particular season? So this is matching the scope in terms of space and time to your question. The next is methods. Are the methods applied in a way so that you can really separate the effects of the element in question in your study design? Or are they potentially confounding factors? Give you an example here. For example, let's say you wanted to attribute the effect to, let's say, a bascular mycorrhizal fungi. But what you have done is actually inoculate plants with a complex inoculum that contains mycorrhizal fungi, but roots, but also other soil microbes. And have you measured then the effects? Then in the end, you cannot really attribute that effect directly to just mycorrhizal fungi, because you've changed more than one thing. Namely, you have added all these other microbes and potentially also the other material that is contained in the soil inoculum. So therefore, you need to always ask, is the method really correctly matched to the element of the treatment that you really wished to make a statement about? It can be very tricky sometimes, but it's essential. Another essential design feature is, has the study been replicated at the right level? For example, if for whatever reason you wanted to compare some feature among grasslands and forests, you cannot just have one grassland and one forest site that you then repeatedly sample and then compare with each other, because that would be pseudo-replication. So if you wanted to make a statement about comparing grasslands and forests, with respect to some feature, for example, soil biogeochemical cycling, then you need to make sure that you have in the landscape replicated, say, grasslands and forest sites with independent management. So it's important to avoid pseudo-replication and to replicate at the right level that matches your question. And then finally, a design feature that's always worth looking into in great detail are the controls. Sometimes you need more than one control. Sometimes the control is much more work than the treatment, because you need to really account for all of the factors that you also change concurrently with your treatment, but that you're not interested in, and the effects that you want to exclude as potentially confounding effects in your treatment. So were the right controls really used in the experiment to really cover all the effects that you could have also changed along with your treatment? That sometimes can be very tricky. So that's all the mostly the design aspects of the study that are very important to look into. The next one is, do the data, like the results that are obtained, really support the statement that's being made? Like, were the right statistics basically being used? In ecology, there is an ever-increasing demand on statistical expertise, and you need to have the right data types, the right data quality, the matching statistical tests to support your statement. Otherwise, your data don't support your statement directly and are therefore suspect. Another important quality feature is the external validity of the study. So was this study being carried out with one plant species under one particular set of conditions using one soil type, or were parameters sampled more comprehensively so that you can say that this particular phenomenon was also valid under a broader set of environmental conditions? So this is the degree to which your results can be generalized. Having a result that has low generalizability is not per se a flaw. It just means, well, your results pertain to that particular study system. But then very often you need to be careful about, in the discussion section, these particular results that are really relevant for this particular system, that they have not been extrapolated unduly to other parameters, for other parts of the parameter space. And it is clearly a very important feature of study quality to have high external validity. So to look at the stability, let's say, of a phenomenon under a range of different conditions that makes this observation more generalizable. And in particular in ecology is very important, because you have different ecosystem types, you have different, very many different situations, and therefore it is highly desirable to have a good external validity in a particular experiment. Another important feature is to look at the literature that's being cited. This is, of course, also difficult to do perfectly, but it needs to at least adhere to certain general conditions. Like, you have a general source to support a general statement, you have a specific source like a primary literature paper to support a specific point that you are making. Is proper credit being given to the first paper to have made a certain point, or are you just citing them, or are they just citing the most recent papers in a field? You should do both, right? You should give credit to the first paper having made some observation, and at the same time you should also, of course, show that you are aware of the state of the art of the literature as well. Or is preference given to certain types of authors that may be just all connected to each other? Is there sort of a cluster in the authors that's difficult to avoid, because you have a natural tendency, of course, to cite the people that you know? But you have to work hard on that as an author and try to get a very balanced and fair account of the literature that is to be cited in that field. And not having achieved that is a problem. And then finally, when it comes to the description of the methods, we already talked about the design, but the level of detail given in the methods is also important. Is it in principle sufficiently detailed so that people could reproduce the results, given that they have basic insights into the methods being used? Are the data basically deposited? Is the code available in a general repository? Very many times this is important, so you can basically reproduce the analyses a particular paper has made. And that is also a mark of quality, basically, that it is transparent on the one hand, and also sufficiently detailed in the description of the method, so that it's in principle reproducible. So that is also an important feature in the end. So you can see that there is very many points to consider. Each of these points that I've mentioned, you could have a whole section on those, and very many times it takes a couple of brains. As in our lab meetings, this is always amazing to me how many things people come up with when they look at a paper from different directions. But of course, you need to, over time, sort of learn to make a judgment on a paper and appraise its value based on a number, at least, of these points, knowing full well that you may have missed something. Just like the reviewers, of course, may have also missed something. This is how this happens, and how sometimes less than perfect papers, of course, get published. This is normal, and it's human. And every time in our lab meeting we have a few dozen people getting together. Only when you have that breadth of input and experience looking at a paper, you can see where there are some potential flaws sometimes becoming apparent that were not apparent, let's say, to me as a single person. But if you keep these various points that we talked about in mind, I think that you will have sort of a very useful checklist of features to check in a paper. Hi there! If you like this video, don't forget to click like down there, and also remember to subscribe to the channel, and feel free to leave comments. See ya!